Use of bacteria. Microorganisms can be used by humans to produce foods and other useful substances. The most common type of microorganisms used in biotechnology are bacteria. They are useful because they are capable of producing complex molecules, e.g., certain bacteria added to milk produce enzymes that turn the milk into yogurt. They are also useful because they reproduce rapidly, meaning the amount of chemicals they can produce can also rapidly increase. Usefulness of bacteria. Extended. In addition to the benefits of rapid reproduction and the ability to make complex molecules, bacteria have other benefits in biotechnology. There are few ethical considerations to growing them in large numbers in the laboratory. They possess plasmids. Plasmids are small, circular loops of DNA which can be an ideal way of transferring DNA from one cell to another during genetic manipulation. Everyday products made with biotechnology. Biofuels. Yeast is a single-celled fungus that uses sugar as its food source. When it respires, ethanol and carbon dioxide are produced and energy is released. The alcohol produced by fermentation of glucose can be used as biofuel. The ethanol produced in this reaction is increasingly being used as a biofuel, a fuel made from living organisms rather than a fossil fuel like oil, coal or gas. In countries such as Brazil, biofuel is partly replacing petrol as the fuel for cars and other vehicles. Plant material is used as the substrate for producing ethanol. As a source of glucose, it is chopped up into small pieces and mixed with yeast, which respires anaerobically and produces ethanol. The liquid is separated from the remaining solids and any water is removed, leaving a concentrated solution of ethanol. Sometimes the waste parts of crop plants, such as the stalks or outer leaves, are used, but in other places, crops are grown specifically to be harvested for making ethanol. In some places, this is causing concern that there is less land available for local people to grow food crops needed for survival. Bread. Yeast will respire anaerobically if it has access to plenty of sugar, even if oxygen is available. This is taken advantage of in bread making, where the yeast is mixed with flour and water and respires anaerobically, producing carbon dioxide. The carbon dioxide produced by fermentation, anaerobic respiration, of glucose is what makes bread dough rise. The carbon dioxide produced by the yeast during respiration is caught in the dough, causing the bread to rise. Fruit juice production. Fruit juice is produced by squeezing the fruits to remove the juice. Chopping the fruit up before squeezing helps to release a lot more juice, but this does not break open all the cells, so a lot of juice is lost. By adding an enzyme called pectinase to the chopped up fruit, more juice is released. Pectinase works by breaking down a chemical called pectin that is found inside plant cell walls. Once pectin is broken down, the cell walls break more easily and more juice can be squeezed out of the fruit. Adding pectinase to fruits also helps to produce a clearer juice, as larger polysaccharides like pectin can make the juice seem cloudy. Once they are broken down into smaller molecules, the juice becomes clearer. Biological washing powders. Many stains on clothes are organic molecules: oil from skin, protein from blood, fat and protein from food. Detergents that only contain soap can remove some of these stains when mixed with hot water, but it can take a lot of time and effort and very high temperatures to remove the stains entirely. Biological washing powders contain enzymes similar to the digestive enzymes produced in the alimentary canal that help to break down large food molecules. Using biological washing powders has several advantages, including quickly breaking down large, insoluble molecules such as fats and proteins into smaller, soluble ones that will dissolve in washing water. They are effective at lower temperatures, meaning less energy and money has to be used in order to wash clothes to get them clean, as washing water does not need to be heated to higher temperatures. They can be used to clean delicate fabrics that would not be suitable for washing at high temperatures. Other uses of biotechnology. Extended. Lactose is the sugar found in milk. Human babies are born with the ability to produce lactase, the enzyme that breaks down lactose. In certain areas of the world, many people lose the ability to produce lactase as they get older. This means that they can become lactose intolerant and react badly to the lactose in milk and products made from milk, cheese, yogurt, etc. Symptoms of lactose intolerance include nausea, flatulence and diarrhea, as their digestive system is upset by the lactose. Milk can be made lactose-free by adding the enzyme lactase to it and leaving it to stand for a while to allow the enzyme to break down the lactose. Lactose-free milk is a product made from adding the enzyme lactase to dairy milk to break down the sugars in it. Penicillin production. Penicillin was the first antibiotic, discovered in 1928 by Alexander Fleming. He noticed that some bacteria he had left in a petri dish had been killed by the naturally occurring penicillium mold. The penicillium mold produces a chemical to prevent it being infected by certain types of bacteria. Penicillin produced by the fungus Penicillium inhibits bacterial growth. The chemical was isolated and named penicillin. Since the discovery of penicillin, methods have been developed to produce it on a large scale, using an industrial fermenter. A diagram of an industrial fermenter used to produce large quantities of microorganisms. Fermenters are containers used to grow, culture, microorganisms like bacteria and fungi in large amounts. These can then be used for many biotechnological processes like producing genetically modified bacteria and the penicillium mold that produces penicillin. The advantage of using a fermenter is that conditions can be carefully controlled to produce large quantities of exactly the right type of microorganism. Condition. Why and how it is controlled. Aseptic precautions. Fermenter is cleaned by steam to kill microorganisms and prevent chemical contamination, which ensures only the desired microorganisms can grow. Nutrients. Nutrients are needed for use in respiration to release energy for growth and reproduction of the microorganisms. Optimum temperature. Temperature is monitored using probes and maintained using a water jacket. This ensures an optimum environment for enzymes to increase enzyme activity and prevent denaturation. Optimum pH. pH is monitored using a probe to check it is at the optimum value for the microorganism being grown. The pH can be adjusted using acids and alkalis. Oxygenation. Oxygen is required for aerobic respiration to take place. Agitation. Stirring paddles are used to ensure temperature, pH, nutrients, and oxygen are all distributed evenly throughout the fermenter. Waste. The contents are filtered to remove waste created by the microorganisms. Mycoprotein. The process of creating food from a fungus. The fungus Fusarium is cultured grown on an industrial scale in fermenters. These fermenters are large vats that can be kept at the optimum pH and temperature for Fusarium to grow. The fungus is grown in aerobic conditions. It is provided with oxygen and provided with glucose syrup as a food source to allow the fungus to respire. The fungus grows and multiplies within the fermenter. The fungal biomass is then harvested and purified to produce mycoprotein. Mycoprotein is a protein-rich food suitable for vegetarians, e.g., it is used in corn TM products. Production of insulin. Genetic modification is changing the genetic material of an organism by removing or altering genes within that organism, or by inserting genes from another organism. The organism receiving the genetic material is said to be genetically modified, or is described as a transgenic organism. The DNA of the organism that now contains DNA from another organism as well is known as recombinant DNA. The gene for human insulin has been inserted into bacteria which then produce human insulin which can be collected and purified for medical use to treat people with diabetes. Genetic modification. Definition. Genetic modification. Genetic modification is changing the genetic material of an organism by removing, changing or inserting individual genes from another organism. The organism receiving the genetic material is said to be genetically modified, or is described as a transgenic organism. The DNA of the organism that now contains DNA from another organism as well is known as recombinant DNA. Genetic modification. Examples. There are many examples of genetically modified organisms, including the gene for human insulin has been inserted into bacteria which then produce human insulin which can be collected and purified for medical use for diabetics. Crop plants, such as wheat and maize, have been genetically modified to contain a gene from a bacterium that produces a poison that kills insects, making them resistant to insect pests such as caterpillars. Crop plants have also been genetically modified to make them resistant to certain herbicides, chemicals that kill plants, meaning that when the herbicide is sprayed on the crop it only kills weeds and does not affect the crop plant. Some crops have been genetically modified to produce additional vitamins, e.g., golden rice contains genes from another plant and a bacterium which make the rice grains produce a chemical that is turned into vitamin A in the human body, which could help prevent deficiency diseases in certain areas of the world. Did this video help you? Yes. No. Genetic modification. Examples. Extended genetic modification using bacterial production of a human protein. The gene that is to be inserted is located in the original organism. For example, this could be the gene for human insulin. Restriction enzymes are used to isolate the required gene, leaving it with sticky ends, a short section of unpaired bases. A bacterial plasmid is cut by the same restriction enzyme, leaving it with corresponding sticky ends. Plasmids are circles of DNA found inside bacterial cells. Restriction enzymes cut DNA strands at specific sequences to form sticky ends. The plasmid and the isolated gene are joined together by DNA ligase enzyme. If two pieces of DNA have matching sticky ends because they have been cut by the same restriction enzyme, DNA ligase will link them to form a single, unbroken molecule of DNA. DNA ligase is used to join two separate pieces of DNA together. The genetically engineered plasmid is inserted into a bacterial cell. When the bacteria reproduce, the plasmids are copied as well, and so a recombinant plasmid can quickly be spread as the bacteria multiply, and they will then all express the gene and make the human protein. The genetically engineered bacteria can be placed in a fermenter to reproduce quickly in controlled conditions and make large quantities of the human protein. Bacteria are extremely useful for genetic engineering purposes because they contain the same genetic code as the organisms we are taking the genes from, meaning they can easily read it and produce the same proteins. There are no ethical concerns over their manipulation and growth, unlike if animals were used, as they can feel pain and distress. The presence of plasmids in bacteria, separate from the main bacterial chromosome, makes them easy to remove and manipulate to insert genes into them and then place back inside the bacterial cells. GM crops. Extended.